Welcome to 2-Minute Anesthesia in Critical Care. Let's discuss three important pharmacokinetic topics: elimination, clearance, and volume distribution. Some of the topics we're going to discuss today are relatively complex, and therefore we'll start with a brief overview. ADME: absorption, distribution, metabolism, elimination. Firstly, absorption. Drugs which are given by the intravenous route, which is common in critical care, directly enter the bloodstream, leading to 100% bioavailability. And therefore the absorption of intravenous drugs is not measured. Once absorbed, it circulates within the systemic circulation and then distributes to tissue. Metabolism may occur, leading to breakdown of that drug, and finally elimination, irreversible removal of that drug from the plasma. There's three important terms which we should be covered: excretion, removed from the body; elimination, removed from the plasma; and clearance, volume of plasma cleared per unit time. These will be discussed individually. Firstly, the volume of distribution. This is the volume of plasma required to account for the total dose of that drug being given, if that drug was only present throughout the body at the same concentration as that found in the plasma. That may sound a bit complicated, but will be discussed a little bit later. It's documented as litres per kilogram, and the volume of distribution is clinically important to determine the loading dose of a drug and estimate blood concentrations in the treatment of overdose. Now let's look at a worked example. So you have a 70 kilogram individual. This results in a plasma volume of between 3 and 3.5 litres. You give an intravenous injection. Some of that drug will bind to plasma proteins, whilst others will distribute to tissues or move into the extracellular fluid. So the volume of distribution is equal to the dose of the drug divided by the measured concentration of the drug. So if you give a 1000 microgram drug and the plasma concentration is measured at 2 micrograms per litre, the volume of distribution is 500 litres or 7.1 litres per kilo. As this 7.1 litres per kilo is greater than the plasma volume of approximately 3.5 litres, there must be greater spread to additional tissues. This can be accounted by different factors of that drug relating to lipid solubility, protein and tissue binding. Drug elimination is the removal of the drug from the body. This can be achieved via excretion of an unchanged molecule or metabolism and then excretion. Excretion usually involves the kidney. For hydrophilic drugs, these are directly excreted, whilst hydrophobic agents may require modification prior to excretion. Three important factors are involved in drug elimination: the drug elimination rate constant, K, the drug elimination time constant, tau, and the half-life. The elimination rate constant is the rate at which a drug is cleared from the body, assuming first order kinetics. So the fall of that drug concentration would occur in a linear fashion, and the elimination rate constant is expressed as a fraction per unit time. For example, 0.2, which is 20% per unit time. This gives an exponential graph. This makes more sense as the plasma concentration of the drug gets smaller. The 20% removed each time will also get smaller. Next is the elimination time constant, which is a reciprocal of the rate constant, 1 over k. This is the time it takes for the process to complete if the initial rate continued. So in this example, look at the graph. We draw a tangent, which is the yellow line, and this is in the steepest point of the curve down to the x-axis. This usually results in approximately 37% of the original concentration. The half-life is the time it takes for the plasma concentration to drop by 50%. It is directly proportional to the volume of distribution and inversely proportional to the clearance. Now let's discuss kinetics. Zero order. The rate of elimination is constant per unit time, whilst for first order, a constant proportion, i.e. percentage, of that drug is eliminated per unit time. As that concentration of the drug increases, the rate of elimination also increases. The elimination rate can be calculated with the following equation, i.e. how much of that drug is removed, meaning physical removed from the body via, for example, kidneys or biotransformation, from that plasma per unit time. So the rate of elimination, milligrams per minute, is equal to the concentration multiplied by the clearance. However, the rate of elimination is going to change with the plasma concentration. Remember, most drugs have first order elimination kinetics. This is why we use clearance, as it's constant regardless of the plasma concentration. Clearance. This is the volume of plasma cleared per unit time. Drugs can be cleared from the body in different ways, for example, organ systems and metabolic pathways. Hepatic and renal clearance are the most important, but other pathways do exist: GI, biliary and pulmonary. The total body or systemic clearance is the sum of all clearance pathways. Let's discuss renal clearance, as this is the most widely used. Renal clearance can be measured in multiple ways. The older method requires urine collection and blood sampling. Renal clearance is equal to urinary drug concentration U multiplied by the urinary flow rate divided by the plasma concentration. Now let's look at a worked example. For example, the urinary drug concentration is 100 milligrams. The urinary flow rate is 1 ml per minute. This can be calculated by using the total urinary volume over a period of time. For example, 60 ml of urine in 60 minutes. And the plasma concentration is 1 milligram per ml. This results in the clearance of the compound of 100 ml per minute, i.e. 100 ml of plasma is cleared of the substance per minute. Thank you for watching.